In this lecture, we'll be talking about protein structure. Proteins are polymers of amino acids. Amino acids are compounds which are named that because they have both an amino group and a carboxylic acid group on the same molecule, as you can see in these schematized structures below. Now, in order to form this long polymer we call a protein, the amino acids have to be linked together, and they're linked to each other via specialized bonds that we call peptide bonds. The peptide bonds are bonds between the amino group of one amino acid and the carboxylic acid group of the adjacent amino acid. Now, this creates a polarity in these proteins, such that we have one end of the protein which has a free amino group, and we call that the N-terminus, and the other end has a free carboxylic acid group, which we call the C-terminus. Now, in all the structures I've shown you before, there was always a group that's called R, and that R represents a bunch of different structures. These are the side groups of different amino acids. Now, in eukaryotic cells, well, in all cells for that matter, there are 20 different side groups that we typically use in amino acids that make proteins. Now, the structure of this R group is very important because it's the structure of this R group that determines the individual properties of each amino acid. These R groups, or these side chains, fall into several different categories depending upon their chemical properties. These categories include aliphatic, which are nonpolar or hydrophobic amino acids, aromatic amino acids, polar amino acids, and charged amino acids, which can be either basic or positively charged, or acidic or negatively charged. We're going to discuss each of these categories of amino acids in turn. The first category of amino acids that we'll talk about is the aliphatic amino acids. These are glycine, alanine, valine, leucine, and isoleucine. You can see that the structure of each of these side chains is shown in the table here. Now, it's important when you learn these amino acids that you learn not only the names of the amino acids, but also the different codes we use for these amino acids. There's a 3-letter code and a 1-letter code for each of them. For example, glycine can be abbreviated GLY or simply G. And that's indicated in the table which you see on the slide. Now, in the aliphatic amino acids, the side chains consist of simply hydrocarbons, and this makes them very hydrophobic. When we say hydrophobic, we mean that these are things that don't interact very well with water. And so they tend to aggregate with other hydrophobic things rather than interacting with polar things or with water. A consequence of this is that the amino acids with these side chains are preferentially found at the interior of proteins, where they can interact with other nonpolar side chains, and not on the surface of proteins, where they would be forced to interact with water. Proline is also an aliphatic amino acid, but it's a special case because it's actually not even an amino acid. It's what we call an imino acid, in which the side chain is bonded back to the amino group, as you can see in the schematic below. Now, protein has some special functions in protein folding, but for the purposes of this lecture, we'll just consider proline as one of the aliphatic or hydrophobic amino acids. The second category of amino acids is the aromatic amino acids. These are amino acids which contain some form of benzene ring in their side chain. This makes them hydrophobic in general, although two of them, tryptophan and tyrosine, are made less hydrophobic by their hydroxyl or amino groups, as you can see in the structures to the right. Now, the unique feature of the aromatic amino acids that maybe will help you remember them is that they have non-intuitive single-letter codes. For example, phenylalanine is abbreviated F, tyrosine is abbreviated Y, and tryptophan is abbreviated W. The next category of amino acids is the polar amino acids. These are serine, threonine, cysteine, methionine, asparagine, and glutamine. These have polar side chains. These side chains are polar because they have either hydroxyl group or amine groups, which allow them to interact very well with water. As a consequence of that, these are the types of amino acids that are typically seen on the surface of proteins, where they interact with their aqueous environment. Now, most of these have intuitive single-letter codes, S for serine, T for threonine, C for cysteine, and M for methionine. But asparagine and glutamine are slightly non-intuitive. Asparagine is abbreviated N, and glutamine is abbreviated Q. The last category of amino acids is the charged amino acids. Each of these side groups has either a positively charged or a negatively charged group. The positively charged groups we call basic side chains. The negatively charged groups we call acidic side chains. Because of their charged nature, these side chains also interact well with water and are found at the surface of proteins, and are oftentimes, because of their charged nature, found in the active sites or the catalytic sites of enzymes. These amino acids are aspartate, which is abbreviated D, glutamate, which is abbreviated E, lysine, which is abbreviated K, arginine, which is abbreviated R, and histidine, which is abbreviated H. Aspartate and glutamate are the negatively charged or acidic amino acids, while lysine, arginine, and histidine are the basic or positively charged amino acids. Now, the function of each protein is determined by its structure or its conformation, the three-dimensional shape that it assumes. And this conformation is unique to every protein and is determined by a lot of different structural interactions. These structural interactions occur at one of four different levels: primary, secondary, tertiary, and quaternary interactions. When we talk about primary structure or primary interactions, we're talking about the protein sequence or the order of the amino acids in the peptide that makes up the protein. And this is determined by the particular peptide bonds that are formed and is ultimately determined by the RNA and DNA sequence from which that protein is derived. The secondary structures are structures that are formed by hydrogen bonds between the amino and the carboxyl groups of individual amino acids. The interaction between these amino and carboxyl groups form different structures that we call alpha helices or beta bends or beta pleated sheets. Examples of these are shown here. In the top you see what's an alpha helix, and you can see that there are hydrogen bonds between the amine groups of certain amino acids and the carboxyl groups of other amino acids. And this causes this peptide sequence to form a spiral or a helix that we call an alpha helix. Now you can see in the schematic below, at the bottom of the slide, we have what we call a beta pleated sheet. And again, these are interactions between carboxyl groups and amino groups which cause two strands of a protein or two lengths of peptide to interact with each other in a parallel or an anti-parallel fashion as shown below. Tertiary structure describes the three-dimensional form that a polypeptide assumes after the secondary structure is already in place. Now this structure is determined by a number of different forces, and these can include covalent bonds. These are things like disulfide bridges, which can occur between two cysteine residues, two cysteine amino acids in a protein. They could also be ionic bonds, which are bonds formed between positively charged and negatively charged groups on basic and acidic amino acids. There are additional hydrogen bonds formed primarily between the side chains of different amino acids in the polypeptide, which would allow it to form a three-dimensional structure. And there are also hydrophobic interactions, which are interactions between hydrophobic or aliphatic amino acids, causing them to be buried in the interior of the protein. That forces the protein to assume a particular structure. Quaternary structure refers to interactions between polypeptides in the formation of multi-peptide protein complexes. And these are where you put two or more proteins together to form a complex of proteins, which are required to have certain functions. The forces that determine the interactions between these polypeptides are the same as those which form tertiary structure, namely covalent bonds, ionic bonds, hydrogen bonds, and hydrophobic forces. Now, like any process, biochemical process, translation can also be messed up, and that leads to particular clinical consequences to patients that have defects in protein structure. The following clinical vignette illustrates one of these. A 3-year-old male presents to the emergency room complaining of severe pain in his legs. His mother reports also that his eyes seem to be quite yellow. This is a family that recently immigrated from Africa. When a physical exam is performed, it reveals that the patient has a fever, has tachycardia, scleral icterus, which means his sclera on his eyes are yellow. He has extreme tenderness in his lower extremities. If you even touch it, it hurts severely. You do laboratory studies. You draw blood from this patient, send it to the laboratory, and they discover a normocytic anemia. He has too few red blood cells. And the red blood cells that he does have an abnormal red blood cell morphology, shown in this slide. You can see that most of the cells here are a nice round structure like normal red blood cells are. But there are some that have sort of a crescent shape or banana shape or sickle shape, which is the term that we use to refer to these red blood cells. So what do you think that this patient has? This patient has sickle cell disease. Sickle cell disease is an autosomal recessive disorder, and it's caused by mutations in the beta globin gene. The mutation which causes this disease leads to a replacement of a glutamate residue at position 6 in the polypeptide, which forms beta globin. That gets replaced by a valine amino acid, which is hydrophobic. The resulting hemoglobin is now called hemoglobin S rather than the normal hemoglobin A. Now, the substitution of this hydrophobic amino acid for what is normally a charged amino acid completely disrupts the normal function of the beta globin protein. And when this structure is disrupted, especially under conditions of low oxygen tension, the abnormal hemoglobin molecules tend to aggregate to one another. And this leads to the typical sickling morphology that we saw in those red blood cells. Now, the situations in which low oxygen tension can lead to this event are things like infection, dehydration, or hypoxia. Now, the sickling of these red blood cells leads to two consequences. First of all, the abnormal red blood cells are very susceptible to lysis and clearance in the liver and spleen, and this leads to a hemolytic anemia, the breaking up of red blood cells. The other thing that happens is that the sickled red blood cells tend to aggregate to one another, and when they aggregate with each other, they can occlude the small capillaries in the microvasculature of patients. This can lead to hypoxia of tissues downstream of this aggregation, and that causes acute pain crises, as what's seen in this vignette, painful priapism, dactylitis, which is swelling of the hands and feet, acute respiratory distress, and/or stroke. Now, when this happens chronically over a long period of time, this can lead to infarction and involution of the spleen. This is a process we term autosplenectomy. This puts patients at risk for infection with encapsulated bacteria like Streptococcus pneumoniae and Haemophilus influenzae. These patients are also at risk for Salmonella osteomyelitis, which is a disease that we see almost exclusively in sickle cell disease patients. A consequence of this is all children less than 5 years of age receive penicillin on a regular basis to prevent these infections. They also receive aggressive vaccination for these diseases. How do we treat a patient that comes with an acute crisis like this patient did? Well, we hydrate them, we give them fluids, and we give them oxygen to try to reverse or to try to prevent further sickling, and we try to control their pain. If the crises are especially severe, it may require exchange transfusion, in which we actually remove the patient's sickled blood and replace it with donor blood, which won't sickle. The other thing that we can do is give these patients a drug called hydroxyurea. Given over time, this increases the concentration of hemoglobin F. This is the fetal hemoglobin that has a alpha-2 gamma-2 structure instead of the normal alpha-2 beta-2 structure. And this helps because, remember, it's the beta globin gene, the beta globin protein that's affected in this disease. This treatment tends to decrease the frequency and severity of crisis. This concludes our lecture on protein structure.